All right, so welcome to our second IMPART Research Reels webinars. We are delighted about the new webinar series that the IMPART team is hosting. Before we get started, I would like to introduce it to you all. For those who may not know, the IMPART is a national research team of multidisciplinary researchers and physicians headquartered in St. John at Dalhousie Medicine New Brunswick, that encompasses 45 academic and clinical partners across 8 universities, as well 25 community and funding agency members, and 15 private sector representatives in 2 provincial government departments. So with our initiative to bridge collaboration between universities and between our members, we created a webinar series that our members between our members and where we can share that is held on monthly basis by the IMPART team investigators and trainees on ongoing and completed research within the network. The objective of this webinar series are to improve networking among IMPART team members, enhance opportunities for future collaborations, and support knowledge translation of research findings. If you would like to nominate a speaker for the upcoming research webinar series, stay tuned for our post-webinar survey, where you can recommend someone who you would like to see presenting for our future seminar series, and also provide a feedback for our team. IMPART is also present on social media, so please follow us to find out more about the research collaboration opportunities we have. For today, I am beyond excited to introduce to you today's speaker, who is Dr. Christie Aguiar, who is a biobank manager at the Dalhousie Medicine New Brunswick. She manages the IMPART BioBank that stores human heart samples from patient participants enrolled in clinical research studies at New Brunswick Heart Centre. Her role involves organizing and supervising all aspects of biobanking and sample processing. She also manages patient databases of biobank samples and liaises with biobank collaborators. Christie has worked as a research coordinator with the New Brunswick Heart Centre, and after she joined Dal Med New Brunswick as a biobank manager. She also has a Doctor of Veterinary Medicine, a Master's of Science, and a PhD in Veterinary Biomedicine. I would like now to pass the floor to Dr. Aguiar.
Thank you, Natalia. I'm just going to share my screen. It's not letting me share my screen until you're done sharing. Okay. All right. Assuming you can see my screen well. Good afternoon, everybody. Thank you for making the time today to listen to our webinar. Today, I shall be discussing everything about the IMPART BioBank, and on the agenda we have, what the IMPART BioBank is, what we do at the IMPART BioBank, why and how we bank tissues, the types of tissues we have, and I shall also explain to you who our friends and neighbors are in our research networks. And I will talk to you about why you should use our samples and what you can do with our samples and who funds us and how to obtain samples from the IMPART BioBank. So this is where we are located here in St. John, New Brunswick on the east coast of Canada. And this picture down here is a true representation of our foggy St. John. And this is an image of the IMPART BioBank, which is located in the St. John Regional Hospital. So the IMPART BioBank is a biorepository of samples for research purposes, serving academic labs at Dalhousie University and affiliated institutions. And we were recently given the CEDARNet certification, which is the Canadian Tissue Repository Network certification in 2020. And we are a biospecimen sourcing partner for researchers worldwide, where we receive, store, process, and disseminate biospecimens, their derivatives, and relevant data. So at the biobank, we collect samples from patients undergoing cardiac surgery. We process the samples. We store them with detailed annotated information. We also have serum, plasma, and red blood cells from volunteer participants. And we store our samples in four freezers. And we also have the ELA immunoassay analyzer and the Bioplex, which is a multiplex immunoassay system. So we are located in close proximity to the Dalhousie Medicine New Brunswick research labs and the St. John Regional Hospital's operating rooms that facilitate quick tissue collection from the operating room and timely processing and storage of human samples. So before I get into the details of the biobank, I just wanted to tell you what IMPART really stands for. And we are a group of researchers with a common interest in inflammation, and metabolism, physical ability research, and research translation. So this infographic will give you some statistics of heart disease in Canada and why IMPART researchers are working in this area. Heart disease is the second leading cause of death among Canadians after cancer, where every hour around 12 Canadians die due to heart disease. So we use biobank samples to ask questions like, why is heart disease different in men and women? And what new drugs can be used to treat heart disease to reduce death rate among Canadians? And how can we improve outcomes for our patients undergoing cardiac surgery? Some of the areas of interest to the IMPART team are obesity, cardiovascular diseases, metabolic diseases, and we work on identifying new biomarkers and therapeutic targets for treatment of various diseases by linking to real-world evidence and clinical outcomes. Some of the risk factors that we collect data on from our patients are blood pressures, HDL, LDL, cholesterol levels, hip and waist circumferences, blood glucose level, as well as social demographic data. So to give you a bit of a historical background, the IMPART BioBank developed out of a collaboration between the DMNB principal investigators and the heart center surgeons who were working on the POPIS study, where we looked at obesity and the impact of obesity on post-op outcomes in patients undergoing cardiac surgery. So the objective of the study was to determine if alternative measures of obesity and fitness capacity could identify patients at a higher risk for adverse outcomes. And the other objective is to determine if biomarkers along with clinical variables have a higher predictive power to identify patients at a higher risk of experiencing adverse outcomes. So some of the reasons why we bank cardiac tissues are listed here. Although animal models are used to study human cardiovascular diseases, there is lack of cross-validation between disease models and real-world outcomes. And preclinical models only address key features of complex diseases in humans. Human heart samples are important for mechanistic studies, drug discovery, and validation of cardiovascular diseases. And the standard processing of explanted hearts ensures the quality and reliability of samples. So this diagram here shows you the process of biobanking, where we have a research question, we write a proposal that is reviewed and approved by the Institutional Research Board of Horizon Health Network, and patients are screened for eligibility. They are prioritized based on certain characteristics like age or sex or type of disease. Then the patients are consented for participation in the study, and they also consent to collection of blood samples and tissue collections. The samples are then processed, labeled, and given a specific study code to protect the confidentiality of patients, and the samples are stored at appropriate conditions. The samples are then retrieved for different analyses, and the patient's medical record is reviewed to abstract clinical data, after which it is linked to data generated from lab analyses. So the data is analyzed and research articles are published in peer-reviewed journals and shared with the public and social media. Finally, various knowledge translation and knowledge management activities are carried out that informs subsequent research efforts. So this illustration gives us a description of what types of samples we have in the biobank. So during the bypass and valve replacement procedure, adipose tissues and the right atrial appendage is collected as a part of the procedure, and these samples are processed and stored in the form of a powder. They are fixed in formalin or frozen in liquid nitrogen. And on the right, you see the blood samples that are collected from our patient participants and separated into serum, plasma, and RBC pellets. Then the clinical data is collected from the patient chart along with socio-demographic data, and it's linked with biomarker data generated from the lab through various techniques like gene and protein analyses. And we are excited about our new project where we will be collecting normal healthy hearts from autopsy patients and organ donors. And these would serve as experimental controls to compare to our disease heart samples. And we will have samples from the left and right atria, the left and right ventricles, the left anterior descending artery, and the interventricular septum. So this flow chart shows the liaison between Horizon Health Network and Dal Med. On the top left of this image, under standard of care, we see that patient is admitted to the New Brunswick Heart Centre. They consent to undergo cardiac surgery, and they have an option to participate in various research studies that are approved by the ethics board. And as a part of the translational research, the patient's medical history and surgical outcome data are recorded as standard of care. And patients who voluntarily participate in research studies have their blood and tissue samples stored at the biobank, which is a central repository for the collection of clinical data and tissues. And the biobank provides the opportunity for translational research by serving as a link between standard of care and basic science research. And at the top right, we see the Cardiovascular Research New Brunswick group, which undertakes cardiac outcome studies, cardiology and cardiac surgery trials, as well as device trials. And this elaborate network feeds into IMPART research labs that are also connected to NBIRDT, which ultimately reports health and economic outcomes. So what can the IMPART Biobank do for you? We offer convenient access to tissues and blood samples with access to the entire clinical profile of patients with cardiovascular disease. And we offer expert biobanking services with ethically sourced specimens that are Horizon Health Network REB approved. And we also provide de-identified clinical patient data based on your research needs. We offer multi-format samples of high quality that are efficiently processed and stored at optimal temperatures. The IMPART Research Lab performs gene analyses, immunohistochemistry, proteomics, metabolomics, and biomarker research to prevent, diagnose, and treat cardiovascular diseases. And some of the reasons why you should use our samples is you have the opportunity to study cardiovascular disease in a homogeneous New Brunswick population, and access to high quality samples from patients with cardiovascular disease, as well as the access to de-identified medical history and complete clinical profiles. You also have the opportunity to collaborate with IMPART researchers when you use our samples. And some of the analyses that you can potentially carry out with these samples are genomic analyses like whole genome sequencing or whole exome sequencing, transcriptomic analyses like RNA-seq or qPCR, proteomic analyses like Western blotting, qPCR, ELISA, and mass spec, or metabolomics like UPLC and multiplex immunoassays. Getting samples from us is a simple process. You would contact the Biobank Manager, you complete a request form, you submit your REB certification, or you request to collaborate with IMPART PIs. You complete a MTA, the IMPART BioBank would then ship the samples to you. Once your project is complete, you return the data and destroy any unused samples. So this timeline shows you some of the work that we have done using biobank samples. So the study began, the OPUS study began in December of 2014, and our first paper was published in 2015, looking at the role of TFAB in obese diabetic patients. Please do watch our previous webinar by Dr. Pulin Kunil for more details on these studies. And following that study, the next publication looked at CD16 expression and NLR ratios in cardiac surgery patients, where NLR emerged as an important inflammatory biomarker. The next study looked at further validation of reference genes for qPCR that was studied in cardiac muscle and adipose tissues. The next publication compared LPA receptors in human biobank samples and obese mice, following which a protocol paper describing the OPUS trial was published. And our next paper looked at fibrosis and atrial fibrillation in older patients undergoing cardiac surgery. And the next couple of studies looked at promising biomarkers like GDF15 and branched chain amino acids in biobanked tissues to potentially identify high-risk cardiac surgery patients. And all of the above publications in the previous slide have contributed to innovation, discovery, and improved patient care, where we have been able to better define risk factors associated with cardiometabolic diseases. And now we have a better understanding of clinical, cellular, and molecular mechanisms involved in the development and progression of metabolic diseases. And our efforts in the long run will be focused on designing and testing new therapeutic strategies to improve outcomes with cardiovascular diseases. And all of this was made possible by DMRF, who has funded our Biobank initiative by the 2019 Molly Appeal grant led by IMPART and the CVRG Wave 2 Dalhousie team. And we have also been funded by the St. John Regional Foundation and the Government of New Brunswick and Heart and Stroke New Brunswick Foundation. And reach out to us if you have any questions or any comments to our social media channels. And thank you for your attention.